when you attempt to answer an exam question on SLE, the difficult part is not about the symptoms or the treatment, but it is about providing an explanation on the cell level and on the molecular level. This is what we're going to explain in this video. We will break down the process in several steps. We will explain them by creating a mind map and by making also notes that will help you memorize stuff that you need to use to answer your question in an exam situation. In systemic lupus erythymatosus, B cells, which are self-reactive, are stimulated to produce autoantibodies against DNA, against chromatin, and against ribonucleoproteins. Let's see how this process actually begins and how. Okay, it all starts with the apoptosis of particular cell types, which is a normal and physiological process. Now, these apoptotic cells, as they die, they secrete signals that reach the tissue the phagocytes are on the tissues. And these signals are called tolerizing for a specific reason that we'll see now. These phagocytes will then attempt to remove the apoptotic cells. The apoptotic cells also, keep in mind that they also produce and release self-antigens. Now, we call these signals from the apoptotic cells as tolerizing because they make the phagocytes promote anti-inflammatory response and also repress the presentation of the self-antigens released by the apoptotic cells to repress their presentation in an activating form. And if they don't do that, then the phagocytes would normally secrete pro-inflammatory cytokines IL-6, IL-12, and also co-stimulatory molecules like B7-1 and induce an autoimmune response, but they don't do that. The phagocytes act as antigen-presenting cells in the presence of the factor of the cytokine TGF-beta. They present the self-antigens to the naive T cells, which circulate in the tissues as well through blood circulation. And in the presence of TGF-beta, these naive T cells that recognize the self-antigens develop into T regulatory cells instead of T effector cells. See, the process this way is blocked. They become induced T regulatory cells. These T regulatory cells now suppress other naive T lymphocytes by secreting IL-10 and TGF-beta to differentiate further into T effector cells that would trigger an autoimmune response. This is what normally happens in tissues which renew physiologically, normally homeostatically, with the cells that die and release the self-antigens that would in other circumstances be treated as a stimulus for an autoimmune response. Now, what happens in an autoimmune disease? Let's see what these self-antigens could be. One type of these self-antigens from the apoptotic cells could be DNA sequence C CpG unmethylated sequence. This is not that common in mammalian DNA. It's more common in bacterial DNA, for example. Now, this unmethylated CpG is recognized by toll-like receptors, in particular by toll-like receptor 9. How this is done? Let's say, let's go back to the apoptotic cells. In the case of extensive cell death, and also when this condition is coupled with inadequate clearance of apoptotic fragments by phagocytes, then when these two circumstances are coupled together, then the B cells with their B cell receptors can bind to these unmethylated CpGs. So you've got a B cell with a BCR, and this can bind to a unmethylated CpG. Where does this come from? This is a self-antigen coming from an apoptotic cell in the case of extensive cell death. And why is that? Why is this an self-antigen? Because in this case, with the extensive cell death, the apoptotic cells are rich in these unmethylated CpG sequences. Rich in unmethylated CpG sequences. Now, once the B cell now binds with its B cell receptor, so CpG unmethylated and the BCR receptor, this is then internalized in an endosomal compartment and where the toll-like receptor 9 is found. And now the unmethylated CpG acts as a ligand both for the B cell receptor but also for the toll-like receptor 9. These toll-like receptors are specific for PAMs, but some of these patterns are also found among self-antigens, like the unmethylated CpG. So make also a note here that the unmethylated CpG can be a pathogen-associated molecular pattern found in bacterial cells, common, but also can be found in mammalian apoptotic cells. Okay, and when this happens, TLR9 then secretes co-stimulatory signals that activate B cells. These are the co-stimulatory signals that activate B cells, and these now B cells can produce anti-chromatin autoantibodies. or they can also act as APCs for autoreactive naive T cells, leading to systemic erythematosus, lupus.